Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel, Pharma World. Today's topic is assigning specifications for APIs and other materials in pharmaceutical manufacturing. Section 11.12 of ICH Q7 prescribes all specifications, sampling plans, and test procedures should be scientifically sound and appropriate to ensure that raw materials, intermediates, APIs, and labels and packaging materials conform to established standards of quality and purity. Specifications and test procedures should be consistent with those included in the registration or filing. There can be specifications in addition to those in registration and filing. Section 11.12 prescribes that all specifications, sampling, test procedures must be scientifically discussed and defined. However, in this video, we limit the discussion to assigning of specifications for APIs and other materials. The guideline says that it should be scientifically sound and appropriate. Scientific means there should be a detailed discussion and justification for each set of specifications on the impact on the purity and impurities of the target product. The discussion should include on carryover and turnover of the impurities into the final product. We should understand what is a carryover impurity and a turnover impurity. A carryover impurity carries forward into the final product as such without any transformation. A turnover impurity is generated when it reacts with other chemicals and convert into a derivative impurity. All this detailed information has to be discussed. This information has to be a part of section 3.2.S.2.2 of your DMF. You may refer ICHQ 11 for detailed information for registration filing of the drug dossier. You can have more specifications other than filed in the dossier. Here, appropriate means the specifications should suit the process requirements. Try to understand these detailed requirements for development of specifications for the materials for pharmaceutical drug substance manufacturing. Section 11.13 prescribes appropriate specifications should be established for APIs in accordance with accepted standards and consistent with the manufacturing process. The specifications should include a control of impurities, example organic impurities, inorganic impurities, and residual solvents. If the API has a specification for microbiological purity, appropriate action limits for total microbial counts objectionable organisms should be established and met. If the API has a specification for endotoxins, appropriate action limits should be established and met. So, section 11.13 prescribes conditions for APIs. Here also it prescribes that the specifications must be appropriate. The specifications should be acceptable as prescribed in various other guidelines which will be discussed in coming up slides. Also, the process should be consistent to meet the specifications. The specifications should include details of organic impurities, inorganic impurities, and residual solvents. If necessary, there should be limits for microbial contamination also. If the API has requirement to evaluate endotoxins, appropriate specifications must be established. Let us see the Control of impurities. Organic impurities may come from raw materials generated during the manufacturing process and or during storage. The origin of organic impurities is from raw materials or generated during manufacturing or even during storage. Then how do you go about? See the path forward. Information on these organic impurities has to be obtained from the list of raw materials used in the process, route of synthesis, potential degradation pathways, or even storage under inappropriate conditions. List of raw materials used in the entire process should be obtained first. This should include all solvents and catalysts also. This is a very first step. Route of synthesis gives a lot of details of impurities formation during the process. The discussion should include on the potential carryover impurities and the turnover impurities. Under certain conditions, the drug substance may itself degrade to give undesirable degradant impurities. This may happen during inappropriate storage conditions also. The justification on the organic impurities should be scientific based on the chemistry of the manufacturing process. As discussed so far, everything has to be scientifically derived and risk assessment made. Organic impurities have to be classified as identified, unidentified, specified, and unspecified. Let us see more details on classification of organic impurities. This is very important for you to know. They are classified as identified, unidentified, specified, and unspecified. The identified impurity is also referred as known impurity, and unidentified impurity is referred as unknown impurity. This is another terminology. An identified impurity is an impurity for which a structural characterization has been achieved. Identified means it is identified with a specific structure. The structure has to be established by several tests like GCMS, HPLCMS, elemental analysis, proton NMR, C13 NMR, etc. IR and UV are also used for identification purposes. Unidentified impurity is an impurity for which a structural characterization has not been achieved and that is defined solely by qualitative analytical properties. Example, chromatographic retention time. Unspecified impurity does not have the structure identified by any of the above tests. It is identified qualitatively. Identifying the impurity with specific retention time in a chromatographic test method could be a possibility. A specified impurity is an impurity that is individually listed and limited with a specific acceptance criteria in the new drug substance specification. A specified impurity can be either identified or unidentified. A specified impurity is the one that is individually listed and is controlled with a specific limit. It can be either identified impurity or unidentified impurity. An unspecified impurity is an impurity that is limited by a general acceptance criteria but not individually listed with its own specific criterion in the new drug substance. An unspecified impurity is limited by a general requirement. You may refer section 5.1 and section 2034 in European Pharmacopoeia for more information. Even USP chapter 476 and 1086 are also useful guidelines for some more relevant information on this. Let us see how inorganic impurities are understood. Inorganic impurities are sourced from usage of inorganic reagents, ligands, and catalysts in the manufacturing process. The source of inorganic impurities is from usage of inorganic reagents, ligands, and catalysts in the process. Inorganic impurities can also generate from usage of inorganic salts, other materials like filter aids and charcoal. Usage of inorganic salts can also contribute to the impurities. Most of the manufacturing processes use the filter aids like high flow supercell, Fuller's earth, etc. for filtration. Charcoal in the form of activated carbon also is used widely in pharmaceutical manufacturing. Inorganic impurities can ingress into the product from these items also. So it is necessary to consider this type of materials for establishing the inorganic impurities in the drug substance. Ingress from the process equipment as heavy metals can be another source of inorganic impurities. Leaching of inorganic elemental impurities from the equipment is another potential source. This is generally established as heavy metals. ICH-Q3D is not in the scope of this discussion. We will discuss the contents of Q3D on some other occasion, but this guide provides a detailed insight into the evaluation of potential elemental impurities. You please go through that for more details. Q3A guideline will provide very detailed insight into the rationale for reporting and control of organic and inorganic impurities. So the ICH Q3A guideline gives detailed rationale for establishing the organic and inorganic impurities in the API. Let us see how the residual solvents are established. Other terminology used for this is organic volatile impurities OVI. Solvents are used as vehicles or medium for the reactions to take place. They do not have any therapeutic value. After the reaction, the solvents are required from each process step. After isolation of the final product, the leftover solvents will be removed by drying at appropriate temperatures in a dryer. Many times the drying is done under vacuum for faster removal and at a much lower temperature. Residual solvents are classified as Class 1, Class 2 and Class 3 based on the risk assessment and permitted daily exposure. The list of solvents are classified into these three classes. The limits are assigned based on the permitted daily exposure. This is done by a detailed risk assessment report. But as a general recommendation, usage of Class 1 solvents in the manufacturing process should be avoided as they are potential carcinogens. But the list which is provided in these three classes is not all inclusive. Any residual solvents which is not a part of any of the classified list, then it is considered as Class 4 solvent and has to be established through a risk assessment report before assigning the limits. The residual solvents are controlled as prescribed in ICH Q3C. USP 467 also provides the same information. So the ICH guideline and the USP guideline provides a lot of details on the residual solvents. The acceptable limits for Class 1 and Class 2 solvents can be tightened to 50% of the ICH limits if they are much below the prescribed limits. For routine monitoring, there is a flexibility for Class 1 and Class 2 solvents. If the residual solvents are much less than the ICH limits, the routine limits could be tightened. Even if you get very less amounts of residual solvents routinely, it is acceptable to limit the specifications to about 50%. For example, if methanol is present in the range of 100 to 200 ppm on a routine screening against the actual limit of not more than 3000 ppm, it is adequate to revise the specifications to not more than 1500 ppm. Acceptance criteria for class 3 solvents need not be tightened, even if it is much below the limit of 0.5%. So for class 3 solvents, in such conditions also you need not tighten the limit of not more than 0.5. Let us see how the identification tests are established. In addition to the above, identification testing should establish the identity and should be able to discriminate between the compounds of closely related structure which are likely to be present. So let us look into other important specification requirements. Identification tests are very important to establish the identity of the material. It should be like a thumb impression specific to a particular person. We know that all of us may look alike, but each is identified specifically by a thumb impression and the iris of the eye. Similar is the case with identity of materials. It should be identified from similar types of compounds. Identity tests should be specific for the new drug substance. Example, infrared spectroscopy. Infrared spectroscopy identification is one of the important tools to identify the compound clearly. Identification by chromatographic retention time may also be utilized to establish the product identity. So chromatographic identification is also a specific test to the compounds under a set of conditions. There can be more than one identification test. As seen in the identification of human being, there are more than one identification test, the thumb impression and the iris. There can be more than one identification. You can notice that there may be up to 4 different identification tests for the API. European Pharmacopoeia prescribes that in all cases, the first identification should be conducted on the article. So if the article has several identification tests classified as first identification and second identification, it is necessary to carry out the first identification test always. Let us see the importance of other tests to be included in the specifications. To make the specifications comprehensive, it is necessary to have the following also. Other important specifications that are required to be included are, say, this is a measure of quantitative content of the active agent in the product. It is closer to 100% in all cases. To accommodate the analytical variations, there could be a close range of the limits. Water content. It is specific for water content. When water is the only solvent that is used in the final isolation of the product, this test is very specific. This has to be carried out by Kalfischer titration method. The USP 921 gives full details on this. You can refer other compendia also for similar information. In such cases, the LOD, the loss and drying test, is not valid. LOD, loss and drying. This test is useful for other organic solvents than water. LOD test is carried out at different temperatures as per the requirements. LOD under vacuum is also there in several monographs. chiral impurities if applicable. This is valid for the same type of chemical structure but spatially arranged in opposite direction. This is valid for chiral compounds only. Polymorphism. This is a physical crystal nature of the compound. This is not valid for amorphous materials. Heavy metals. The information discussed in slide 7 is valid here for heavy metals. There is a detailed strategy to delete USP 231 and implement USP 232 and 233 with reference to the risk assessment and evaluation of the elemental impurities as per ICHQ 3D. But discussion is limited up to this only right now. We will focus in the coming up opportunities on ICHQ 3D. Microbial limits. It is also necessary to establish the freedom from bioburden wherever applicable. There are some physical attributes to be included in the specifications. So, following additional physical attributes are also recommended to be included in the list of specifications. Description, odor, texture. Description is also referred as appearance. It is very important to check USP 631 for this test. The test method should include the intricate details. The section prescribes the perception of color and color matches is dependent on the conditions of viewing and illumination. Determinations should be made using diffuse uniform illumination under conditions that reduce shadows and non-spectral reflectance to a minimum. The surface of powder should be smoothed with a gentle pressure so that the planar surface free from irregularities is presented. Liquids should be compared in matched color comparison tubes against a white background. If the results are found to vary with illusion, those obtained in natural or artificial daylight are to be considered correct. Instead of visual discrimination, a suitable instrumental method may also be used. See how much content is there in that chapter 631. The test may look very simple. Description, odor or texture. It is very important that you have to go through this chapter of USP 631 fully. Particle size less than 75 microns. This test is generally done using a laser particle size analyzer for particle sizes less than 75 microns for more accurate results. Bigger particles sized materials can be tested by sieving test. You can refer USP 786. That gives a great detail on this sieve test for more than 75 microns. Bulk density. This test is an important aspect where compressibility is a process requirement. This test includes apparent density, tap density, etc. USP 616 provides a lot of details in this. Surface area. This is a rare test used for excipients like magnesium stearate. This decides the flowability of the formulated granules for getting the required quality of the tablets. I hope you understood the requirements for establishing the specifications for APIs and other materials used in the manufacturing process. Thanks for watching. For more videos, please do subscribe. like, and share. Thank you.